Right now, living inside your digestive tract, there are approximately 38 trillion bacteria. More bacteria than there are human cells in your entire body. They weigh roughly 1.5 kilograms. They contain 150 times more genes than the human genome. And until about 15 years ago, science largely considered them passengers, commensals tolerated by the body, performing some digestive functions but fundamentally irrelevant to anything that happened above the neck. That assumption has been overturned so completely and so rapidly that some of the researchers who spent decades studying the gut microbiome describe the last 15 years as the most disorienting paradigm shift in modern medicine. Because what those 38 trillion bacteria are doing is not digesting fiber, not just producing vitamins. They are producing neurotransmitters. They are regulating your stress response. They are communicating directly with your brain through the vagus nerve, through immune signaling, through the bloodstream, and through a dedicated enteric nervous system that contains more neurons than the spinal cord. And the specific composition of those bacteria, which species are dominant, which are absent, which are in what ratio, is associated with your anxiety levels, your depression risk, your stress reactivity, your social behavior, and in several documented studies, your personality traits. Your doctor has probably never asked about your gut microbiome in the context of your mental health, but researchers at the APC Microbiome Institute in Cork, at the Salk Institute, at the Karolinska Institute in Sweden, and at dozens of other institutions are now treating the gut as a second brain, not metaphorically, but anatomically and functionally. And what they are finding is changing everything about how we understand where personality comes from, how mental illness develops, and what you can actually do about it. Today we are going through the science, mechanism by mechanism, because the bacteria living in your gut right now are influencing the way you think, feel, and behave in ways that are documented, specific, and, this is the part that surprises most people, bidirectional. You are shaping them as much as they are shaping you. Before we get into what your gut bacteria are revealing about your mental health, you need to understand the gut-brain axis, the communication infrastructure that makes this influence possible. Because without understanding the highway, the traffic makes no sense. The gut and the brain communicate through 4 primary pathways. The first and most direct is the vagus nerve, the longest cranial nerve in the body, running from the brainstem through the neck, chest, and abdomen to the gut. Approximately 80% of the signals traveling through the vagus nerve are ascending, going from the gut to the brain, not the other way around. The gut is not waiting for instructions from the brain. It is sending a continuous stream of information upward. The bacteria in the gut influence the signals sent through the vagus nerve by altering the chemistry of the gut environment, the metabolites they produce, the inflammatory signals they generate, the neurotransmitter precursors they release, all of which affect the vagal afferent neurons. The second pathway is the immune system. Approximately 70% of the body's immune cells reside in or around the gut. The bacteria in the microbiome are in constant interaction with these immune cells, regulating the inflammatory tone of the entire body, including the brain. Neuroinflammation, inflammation within the central nervous system, is now recognized as a primary driver of depression, anxiety, and cognitive decline. And the gut microbiome is one of the primary regulators of the systemic inflammatory state. The third pathway is the endocrine system. Gut bacteria produce and regulate the production of hormones and neurotransmitter precursors in the enteric nervous system, the 500 million neurons lining the gut wall. Approximately 90% of the body's serotonin is produced in the gut, not the brain. Gut bacteria regulate the enterochromaffin cells that produce this serotonin. They regulate GABA production in the gut. They produce short-chain fatty acids that cross the blood-brain barrier and influence neural function directly. The fourth pathway is the bloodstream itself. Bacterial metabolites, including short-chain fatty acids, enter the systemic circulation and reach the brain, influencing neuroinflammation, neuroplasticity, and the blood-brain barrier's integrity. This is not a simple or indirect connection. It is a 4-lane highway carrying continuous, bidirectional, biochemically specific information. Depression is where the gut-brain connection has been most extensively studied, and where the findings have been most paradigm-shifting. A 2019 landmark study published in Nature Microbiology analyzed the gut microbiomes of over 1,000 individuals from a Belgian population cohort and cross-referenced microbiome composition with depression diagnosis and quality of life scores. 2 bacterial genera, Coprococcus and Dialister, were consistently depleted in individuals with depression, even after controlling for antidepressant use, which is known to alter the microbiome independently. The researchers also found that the gut microbiome's capacity to synthesize the dopamine precursor DOPA. The researchers also found that the gut microbiome's capacity to synthesize the dopamine precursor DOPA was correlated with quality of life scores across the entire sample. The bacteria were not just correlated with depression. They were producing the neurochemical precursors that the brain uses to regulate mood. The findings were extended and replicated in a 2022 study in Nature Communications, analyzing microbiome data from over 3,000 participants in the Netherlands. 13 bacterial taxa showed consistent associations with depression symptoms across the sample, with specific species from the Lactospiraceae family, bacteria that produce butyrate, a short-chain fatty acid, with potent neuroprotective and anti-inflammatory effects, showing the strongest inverse associations with depressive symptom severity. The causal question, whether the microbiome changes cause depression or depression causes the microbiome changes, has been partially answered by germ-free animal studies, one of the most compelling experimental paradigms in microbiome research. Germ-free mice raised without any gut bacteria showed dramatically elevated stress responses, anxiety-like behavior, and abnormal social behavior compared to colonized controls. When these germ-free mice are colonized with microbiomes from depressed human donors, they begin showing depression-associated behaviors. When colonized with microbiomes from healthy human donors, these behaviors do not emerge. The microbiome is not just correlating with mental state, it is producing it. A 2022 randomized controlled trial published in Nature Mental Health, the first RCT examining probiotic supplementation for major depression, found that adults with major depressive disorder who received a specific multi-strain probiotic for 8 weeks showed significant reductions in depression scores compared to placebo, with the improvement correlated with measurable changes in microbiome composition and in circulating inflammatory markers. The bacteria were moved. The depression moved with them. Anxiety disorder shows similarly robust microbiome associations with increasingly well-understood mechanisms. The gut microbiome regulates the HPA axis through multiple pathways. Short-chain fatty acids from gut bacteria influence HPA axis sensitivity during early life. Germ-free animal studies show that absence of gut bacteria during early development produces permanently elevated HPA axis reactivity that persists even after subsequent colonization. The window during which the microbiome calibrates the stress response appears to be early childhood, making the early life microbiome one of the most consequential developmental influences on lifetime anxiety risk. A 2015 study in Brain, Behavior, and Immunity found significant microbiome differences in generalized anxiety disorder patients versus healthy controls, anxiety patients showing reduced bifidobacterium and lactobacillus, bacteria producing GABA precursors. A 2019 meta-analysis in General Psychiatry, covering 34 controlled trials, found probiotic supplementation significantly reduced anxiety scores. Social behavior and personality represent the frontier of gut-brain research, where findings are newest, most surprising, and most paradigm disrupting. In 2019, researchers at the Salk Institute published a study in Nature showing gut microbiome composition in mice influenced social behavior, with germ-free mice showing dramatically reduced social interaction. The deficit reversed when germ-free mice were colonized with normal microbiome, specifically when oxytocin levels in the hypothalamus were restored, which the researchers found were regulated by microbiome-derived signals. The bacteria were regulating the neurobiology of social bonding. In humans, a 2021 study in Human Microbiome Journal found significant correlations between gut microbiome diversity and Big Five personality dimensions in over 600 adults. Higher microbiome diversity was associated with lower neuroticism and higher openness to experience. Lactobacillus reuteri, which produces oxytocin-modulating signals, was inversely correlated with social anxiety scores. From over 1,000 adults found specific bacterial taxa associated with personality traits after controlling for diet, age, and other confounders. Bacteroides species showed associations with extroversion. Prevotella species showed associations with agreeableness. These are not large effect sizes. but they are consistent and reproducible findings that suggest the microbiome is contributing to the biological substrate of personality in ways that were completely unimaginable 15 years ago. What can you do about it? Because understanding that your gut bacteria are influencing your mental health in ways that were completely unimaginable 15 years ago. What can you do about it? Because understanding that your gut bacteria are influencing your mental health. A 2021 randomized controlled trial in psychosomatic medicine, the Diet to Help Depression trial, found that young adults with depressive symptoms, randomized to a Mediterranean-style diet, showed significant depression score reductions compared to social support controls after 12 weeks. The microbiome of the dietary group showed specific compositional changes, increased lactobacillus and bifidobacterium, reduced pro-inflammatory species, that correlated with degree of symptom improvement. Dietary fiber is the primary fuel for butyrate-producing bacteria. A 2020 study in Gut found that every 10-gram increase in daily fiber intake was associated with measurable increases in microbiome diversity and Lactospiraceae abundance, the butyrate-producing family. Fermented foods increase microbiome diversity through direct introduction of live bacterial cultures and reduced 19 inflammatory markers compared to a high-fiber diet alone. Chronic stress, sleep deprivation, and ultra-processed food consumption, all covered in this series, each independently reduce microbiome diversity and shift composition toward pro-inflammatory species. The interventions we have discussed across every video in this series are not separately addressing separate systems. They are all addressing the same integrated biological system from different entry points. Here is what I want you to carry out of this video. The 38 trillion bacteria living in your gut right now are not passengers. They are participants. They are producing the neurotransmitter precursors your brain uses to regulate mood. They are calibrating the stress response you walk through life with. They are regulating the inflammatory environment in which your neurons function. And the specific composition of that microbial community, shaped by your diet, your stress levels, your sleep, your antibiotic history, and the microbiome you were colonized with at birth, is associated with your anxiety levels, your depression risk, your social behavior, and your personality dimensions in ways that are now documented in peer-reviewed literature across dozens of independent research cohorts. The gut-brain axis is not a metaphor. It is a 4-lane highway that your bacteria are driving right now, in both directions, carrying signals that are shaping how you think, feel, and behave every single day. What does your diet look like? How much fiber? How many fermented foods? Drop it in the comments. Tell me where your microbiome is getting its fuel from. I read every single one. Subscribe to the channel because next week we are going deeper into the specific dietary strategies that the research shows most powerfully reshape the microbiome in directions that support mental health. And I will see you there.